Sore throat is common among children. It's usually caused by common cold or flu, and it gets better in its own. But strep throat is different. It is caused by a bacteria called streptococcus and needs to be treated with antibiotics straight away. There are more than 200 different strep bacteria, which causes approximately 700 million infections worldwide every year. In most cases, these infections are mild and can be effectively treated with antibiotics. However, if these infections are left untreated, this can lead to serious complications such as rheumatic fever, which affects the heart, brain, joints, and the skin. Australia has the highest prevalence of rheumatic fever worldwide, mainly among indigenous communities. These serious complications are diagnosed solely based on clinical signs and symptoms, which were proposed in 1944, and there is no laboratory test available to detect these serious complications at their early stage. Rheumatic fever is not directly caused by the bacteria itself. Rather, it is caused by antibodies and immune cells that are trying to kill this bacteria. These strep bacteria has some proteins which are closely resembles the proteins of our heart and the brain. Therefore, the antibodies produced by our immune system not only destroys the bacteria but also our own tissues. Rheumatic fever and rheumatic heart disease are 100% preventable diseases. But we believe an effective vaccine against strep bacteria could address a huge unmet public health demand and controlling of rheumatic fever, rheumatic heart disease, and other diseases caused by the same bacteria. For that, we need to know the exact mechanism which lead to heart and brain damage. These studies cannot be done in human, as it involves two major vital organs, the brain and the heart. Therefore, we developed an animal model which resembles the human disease. We injected rats with these strep bacteria and looked at how it affects the brain and the heart. From my study, we have identified some specific proteins from the heart and the brain which are targeted by these strep bacteria. In addition to that, blood from these rats injected with strep bacteria and patients with rheumatic fever, we have identified some specific protein sequence which are common among different strep bacteria. Since this protein sequence is common among different strep bacteria, we can use this strep protein sequence to develop a diagnostic tool for the early detection of these serious complications. Furthermore, a vaccine is considered a feasible and cost-effective solution to control the diseases. Our studies will accelerate the progress of strep vaccine development to control or prevent more than 500,000 premature deaths due to rheumatic heart disease every year. Thank you.